Welcome to today's webinar, Automating Ancillary Processes, Closing the New Bottleneck in Cell and Gene Therapy Manufacturing. This is brought to you in partnership with Invetech. For those of you who don't know me, hello, I'm Małgorzata. I'm the head of content here at Facilitate, and I will be your webinar host today. Today, we're talking about closing processes around media formulation and viral vector packaging. We all know that these are time-intensive and expensive activities if done manually. We have three fantastic panelists lined up for you today, and David Hoddle from Invetech will be moderating this discussion. So before we jump in, a quick reminder that this is an interactive discussion, so all the questions are very much encouraged. To submit anything, please use the question function on your control panel, and we will address these as they come in. So without further ado, I'll hand straight over to our moderator. David, take it away.
Great. Hey, thanks so much, and welcome everybody to our webinar today. As we said, we've got three great panelists. We have a lot of content that we want to cover today. Really interesting subject, I think, as we think broader around scalable manufacturing as more and more companies are getting, more and more organizations are getting to a place where they are thinking about, okay, how do I take that next step to manufacturing? So with our panel today, I want to introduce really quickly our panelists. This morning we have Amy Shaw. Amy is the engineering and automation lead for cell and gene therapies at Takeda. Randy Schweiker is with us from BMS. Randy is the director of engineering and automation at BMS. And we have Fareed Ingma as well. Fareed is the senior director for cell and gene therapy manufacturing at GSK. So with that, very quickly, I want to thank the organizers, thank the team at Facilitate for providing this opportunity for us to have this webinar this morning. And I think we're going to jump right in. So when we think about the entire cell and gene therapy manufacturing process, we think about the core manufacturing process, but also think about processes that are sort of outside that core. I'd like to get a definition of what you all think ancillary systems encompass. And Randy, let's start with you on that.
Thanks, David. Yeah, so I think that we all have a pretty good idea of what we're talking about when we say ancillary systems. We understand that there is this core process. This is for autologous anyways. It is the process whereby the material from the patient is processed through unit operations to that final fill unit operation. Anything beyond that's outside of that allows us to prepare material and systems to feed into that core process is what we think of as ancillary processing. And then in terms of viral vector, the development, the production of the viral vector itself is a core process, if you will. But we also would like to include the filling, if you will, of that, the preparation of that material for use in the core process also is an ancillary operation, because it again feeds into our core process, and that's what we're trying to capture into this ancillary processing arena.
Okay, and Amy, how does Takeda define ancillary systems?
Yeah, absolutely. I mean, I couldn't agree with Randy more. I think for us, focusing on the cell therapy aspect, regardless of it's allogeneic or autologous, anything that's outside of those initial cells really and being added into those cells, we would consider ancillary. So while they could be core processes on their own, what we're bringing into the cell therapy process is considered ancillary.
Okay, and Fareed, any differences or anything that GSK views as sort of different around ancillary systems?
No, we are pretty aligned here. I echo what Randy and Amy said. It's, we define it as any input or any material that goes into that core process. So think here media, any additional reagents, and that includes the polishing steps, the fill finish, etc., at the end of the process as well.
Mm-hmm. Okay. And so we think about those processes as sort of outside of that closed, integrated, automated core manufacturing system or manufacturing process. When you think about closed versus integrated versus say automated, are those in your minds, are those complementary, or how do you view that? And I think with this, I want to start with Fareed.
Sure. So they're certainly not, they are certainly complementary, but not necessarily needs to happen at the same time. The closer you get to that sterile product, the closer you get to that drug product in autologous cell and gene therapies, the more likely these three aspects are to be connected. That's the automation, closure, and digitalization of the data that comes out of your system. This is tied to the vein-to-vein time that's critical in autologous cell and gene therapies. That's the time that it takes to get the cells from the patient and then return them to the patient at the end of the journey. And making that as short as possible is a key driver in autologous cell and gene therapies, because the patients that we're treating are typically really sick, sometimes on their fourth line of therapy in the clinical setting. So it is very important to turn around that information quickly, and the more you do in reducing that release cycle time, the better you are off in getting that material back to the patient in a timely manner. And again, whether you're looking to reduce human error, that's the automation piece, reduce the impact of environmental monitoring, that's the closure piece, and get close to that review by exception, that's the digitalization aspect, the better you are in terms of robustness and in terms of reducing that cycle time.
Okay, it's interesting. I'm really glad that you touched on the digital piece of that too, and I think that's something that let's make sure we remember that, because I think we want to hit on that kind of later in our discussion. Amy, when you think about those processes, what do you think about closure, automation, integration? Is that all sort of the same thing, or how does Takeda think about that?
Yeah, I mean, I like to think about automation and closure working hand in hand, taking things out of operators' hands and putting it into automated equipment really gives the opportunity for some of those movements of product or media or any component of the process to be done in a controlled manner. And within that, you could leverage sterile tube sets or anything like that to really contain the system and close it and provide that extra sterility around your product as well.
Okay. Randy, your thoughts on this?
Sure. I think that from the BMS perspective, I think if we asked the group what was the most important thing, they would say closure is the most important thing. And the reason is because with closure, you're able to really make your plant design much more efficient. And so I do think of them as complementary, the closure and the automation. So what I think of in transformation from an automation perspective is how do I maintain, or in some cases introduce closure through the use of my automation. And so in our first manufacturing facilities, which were based upon early clinical manufacturing techniques, it was the closure itself that made the biggest difference in terms of how efficient we were and the improvement of the manufacturing process. And then the automation aspects, especially in the ancillary space, was that next step to indicate that, hey, we can do this even more efficiently if we take what we've done a really good job with in the core processing and start to apply those principles to the ancillary process as well.
Mm-hmm. So, Randy, I'll follow up a little bit with you on this one first. So when you look at those processes, when? I mean, I guess the question is, okay, you're starting off with these as sort of manual, and you may have open operations in there. You may be operating under a hood. What's the trigger point? When do you look at this and say, okay, now we really need to automate this part of the manufacturing process, or we need to close and integrate this part of the manufacturing process?
Okay, I'm gonna be an automation engineer and say as early as possible. And the reason I say that is because especially in this field where a lot of the work tends to be developed in early clinical settings, just showing feasibility of the modality or the product. Scientists, and they're not, and clinicians, they're not thinking about the manufacturing space. What is that future? They're thinking about the onesie twosie clinical setting, making sure that they have a viable product and so forth. And they don't tend to think about the future when you go from onesie twosie to hundreds to thousands, and even potentially to tens of thousands, depending upon the particular indication. And so that's where you actually need the engineers and the facility designers and folks who really think about the future of what manufacturing is, to try to get involved as early as possible. Now, I'm going to answer your question, I think, more appropriately by saying, certainly at the stage when you start to develop a concept for manufacturing, right? When you start to design your facility, the concept of how you would like to automate that, that's when you're kind of communicating to your scientists, hey, think about what this looks like once we get into manufacturing, because now we're designing a facility. We have to make some decisions about what this is going to look like. Assume we have some automation associated with these different aspects. The advantage we have at this point in time versus where we were even just a couple of years ago is that we have examples of these kinds of technologies that we can show to the scientists and say, this is kind of what to expect. And so in our case, now that we have some platforms, some automated platforms, the people who are, the scientists who are bringing the next pipeline product along, they can assume that we have those capabilities. They don't have to be, they're not working from the blank slate that we were when we first started.
Okay, great. Amy, thoughts from Takeda's perspective on that?
Yeah, absolutely. I mean, I agree again with Randy. As you start thinking about your facility design, it becomes extremely important to think about how each of the different components that are being brought into your suites are packaged and everything that happens upstream to them. So I think it's a problem in the field to really be balancing when to incorporate automation into any process based upon where it is in the clinic and everything else. But as we think about manufacturing scale up into commercial, you want to push as much as you can into some of those lower grade spaces and reduce the need for really locked down manufacturing. And so if you can bring in ancillary materials that are closed and clean and in a really good condition into your core process, it really relieves some of those constraints as you look at your core process itself.
Okay. And Fareed, knowing a little bit about what GSK is doing from the manufacturing side, and thinking about timing there, what were there some unique challenges that you were thinking about as far as when to begin to close and integrate and automate your ancillary processes?
Yeah, so for us at GSK, we started thinking about, we link, I think everybody in the industry does this, we link automation to process robustness. Closure is also part of that, not part of automation, but process robustness envelope in general. So process robustness is as important in the clinical space as it is in the commercial area. The challenge for us in determining when to invest money in automation, in closure, et cetera, was really linked to the availability of a platform process. Especially in early phase and moving into that mid-phase space, R&D space. The more your processes are platformed, the more likely and the more value there is in investing in that automation and closure as early as possible in the R&D life cycle. That way, if there is a new asset, it becomes more of a plug and play, as opposed to one-off processes where you may need to maintain flexibility. So The challenge for us was to, to lock down that platform process and understand the parameters that could change inside of that box, to put it plainly. And that allowed us to design and think about the areas that we wanted to automate and close a bit more clearly.
Okay, great, thanks. I want to give the audience a heads up. We have actually planned on putting a few polling questions in in the flow of the conversation here. So this is the time when I think the first polling question is going to pop up, and this, again, it's an opportunity for you, the audience, to be pretty interactive here. And while you're viewing that, we are going to continue the discussion. So when thinking about that and thinking about the timing of really closing and automating or automating the process, think about the challenge and what were some of the impacts of that challenge of going to a more closed, integrated, automated process for your ancillary systems. And with this one, let's start with Amy.
Yeah, I mean, there's of course the challenge of when to automate, but I think there's always that challenge across the field also of finding the appropriate automation tool to automate those different pieces of the process. So, cell therapies already has a lot of unique automation, but even looking across different therapies, different cell types, et cetera, you might need different sorts of automation, and choosing the right one for your cell therapy can be difficult. You can look across the market and look what's already out there, but that's not always something that already exists. And so you have to find development partners to build those pieces of equipment, those disposables, et cetera. And so, it gives a lot of space for developing the appropriate piece of automation for your process. So
a quick follow-up there, because you brought up an interesting point about handling different cell types. Are there some unique considerations around that when you were making some decisions and thinking about the challenges you had to overcome, thinking about, say, the characteristics of those different cell types?
Yeah, absolutely. I mean, given the cell type too, it can be different scales, different sorts of volumes that you're working with. Different size cells need different sort of mixing components, et cetera. And so all of those different things have really pushed us towards looking at different pieces of equipment across the landscape, really.
Great. Randy, your thoughts?
Yeah, I think the key aspect that we found was that our autologous processes were the ones where we were much more difficult finding the appropriate automation for the ancillary preparation and so forth. And this is to be expected. I mean, the antibody world, protein production world deals with large bioreactors, large downstream purification equipment, and so forth. The uniqueness of autologous therapy is that the primary process, the core process, is a single patient. You tend to be dealing with much smaller volumes. And so what we found was that there was just not very much automation that was specifically designed for that kind of space, whether that is the kinds of materials that were necessary, the manifolds, the sizes of the bags for holding materials, interfaces, the size of the interfaces of the materials, aseptic connectors and welding lines for welding and so forth. And so that was our biggest challenge, was you realize that we had to get into a design build arena to get these kinds of automation systems developed appropriate for our autologous space. So I think that was one of our biggest challenges.
Interesting. And Fareed, I'll throw it to you for GSK's perspective on this.
Yeah, Amy and Randy touched upon the most important challenge that we run into in our autologous work within GSK, and it's around availability of systems and components that cater to smaller volumes, Think here smaller bags, smaller tubing IDs, etc., that work for us in the autologous cell and gene therapy space when you're talking about, you know, MLs as opposed to the biopharm space. And also the accuracy that you need at that small a volume or that small of a scale. So in terms of trade-offs, you can design a system that fills vector into bags at 5 mLs fairly accurately, but if your process improves and now your efficiency is a bit better, your transduction efficiency is a bit better, and you can drop that volume down to 5 ml or to 1 ml, the accuracy may not be as good. So you end up thinking of ways around that, you know, by still using that 1 ml but diluting it to 5 ml or other to kind of make the system work. Vendors are catching up. They're starting to think about you know, what is needed in this space, but we're not there yet for sure.
Okay. Yeah, interesting. Yeah, hey, I just want to comment really quickly too, because the poll results have popped up, and very interesting. And I think you probably, you know, I think everybody's probably thought about this as the top two. You know, media formulation and prep, and viral vector preparation are really the top two processes that our audience is saying are their bottlenecks. So yeah, very, very interesting. Thanks everybody who participated in the poll there. So moving forward a little bit, and again, kind of bouncing off the last question here, what are considerations that, you know, when you think about raw materials coming in, what are some of the challenges that you have with those raw materials, and what sort of message would you send? I don't know, Fareed, you kind of started this, but what sort of message would you send to your suppliers of raw materials as you continue to, you know, close and integrate and ultimately automate these processes? Let me start with Fareed on that.
Yeah, so again, the challenges are really finding off-the-shelf supplies that we can use directly without having to aliquot under a BSC and an A and a B, or find some kind of closed system to, you know, to make that work. The industry is used to the volumes that the biopharm space, you know, sees, or perhaps even the blood industry to a certain extent, but, you know, is just getting their toe into the cell and gene therapy space, autologous specifically. I think allogeneic and, you know, and AAVs, et cetera, are probably in a bit of a better space, a better place. The message that I would send is, you know, start to think about automating the systems at the vendor and starting to provide, you know, closed systems, smaller volumes, tubing sets that work for us, both from a, you know, an ID perspective, but also, you know, the material of construction. That's also a challenge, because you're linking to 1, 2, maybe 3 systems that are prevalent in the autologous cell and gene therapy manufacturing space, and you really need to cater to that. Start to think about, you know, increasing capacity, and start to think about how you would deliver this, because as Randy stated, this is going to start, you know, volumes are going to start going up very quickly. That's a good thing for the industry, and obviously for our patients. But we need that you know, industry you know, backing to make sure that we can deliver.
Interesting. Randy, your thoughts?
Yeah, it is by far the biggest issue that we have to deal with. Again, you know, the uniqueness of the space in cell therapy and cell and gene therapy is that you have unique reagents. These are not your typical throw it in a big vat, mix it together, and there you got your you know, cell culture media, right? You typically have very unique activation reagents, transduction systems. You know, these all require that you use unique materials and these unique materials have to efficiently be incorporated into your core process, and that is a huge challenge. Even very standard materials have to be incorporated into, again, what Fareed and Amy have already talked about, the small scale that that you have with autologous, and in doing so, you have to have the right kind of format for that material. What we found is that the typical kinds of formats that are appropriate for other, especially clinical scale manufacturing, are really detrimental for manufacturing scale manufacturing. And so the key is working with vendors to get them to better understand what is needed. And I would say in this kind of interim space, and I will call where we are at BMS as an interim space, having the ability to do the packaging ourselves, the repackaging, the aliquoting, and so forth, is valuable. We want that to eventually to be, by our vendors. We want to get from our vendors the materials in the right formats that can be directly incorporated into our core processing. But having the flexibility at this point in time, again, as the vendors begin to take on the responsibility of reformatting the materials appropriately, having that flexibility is very valuable to us. We definitely want to transition as soon as we can, but vendors, they're realizing what space they're getting themselves into in this autologous scale processing, and it's going to take a while to get that transition to occur.
Mm-hmm. Amy, your thoughts?
Yeah, I mean, I agree with the panel here. I think that we're at an interesting point here. Cell therapies is getting bigger and bigger, but we're still using things off the shelf that are coming in bottles. I mean, everyone kind of indicated that media formulation is an issue for them, and things like serum coming in bottles, glutamine, etc. Cytokines are a huge issue for us. They're coming in small aliquots that you have to manually pipette a lot of times. So thinking, looking at the vendors, looking at what we use, if things could come in easily weldable or things that we could transfer into closed containers in a more sterile fashion would be great, I think, for the field, whether it comes in transfer bags, again, with the right ID/OD on the tubing, or even I know some vendors have started providing cytokines already reconstituted in syringes. All of those things are helpful to us on this other end. We'd like to see more and more of that come from the vendors.
Okay, well, and I mean, you hit on something at the end of your comments that I want to kind of go back through with everybody, is a message to the vendors, that yes, packaging is important, and how you scale and how you're thinking about the process you're going into. Are there collaborations that have worked really well, or what are some of the challenges that you faced in going to the supplier community and saying this would be great? Is it standards? Is it other considerations? And I'll start with you, Amy, and just go right back down our panel.
I think that for some materials, it can be the stability. So whether you're changing the packaging or, in the case of cytokines, if it's lyophilized or in liquid, the stability becomes a concern. And so there's a whole nother data package that might have to be created around that. So that can be some of the pushback that we see. But again, that's something that we would like to help build over time so that it can become more standard in the industry.
Okay. Randy?
Yeah, exactly. I think that it's the demand. If there's a demand for the materials and the formatting that's appropriate, they will provide it. And so that's our biggest challenge, is that especially if you're first, second, third in line, or to create or to demand a particular item, you may have to be the fifth or the sixth before they say, hey, you know what, there's a market for this stuff. And so I think that's primarily what the stability, clearly that's core. You have to have the stability in the formats that you're looking for. But I would say in terms of where, what's going to convince the vendors that, this is what we need to provide, it's demand. So the more companies that get cell therapy and autologous in particular, on the market, the more the understanding that this is a modality that's going to be here, that the industry has to provide for, and I think that's going to improve with time as they see that demand increase.
Mm-hmm. And Fareed, your thoughts on this?
Yeah, I thought of something witty to say, but Randy and Amy stole my thunder on this one. I fully agree.
Sorry.
No, joking here. It's the stability and shelf life of the material, and also the volume squared, volume of the material that we're trying to manufacture there, but also the volume of drug product or asset flow that we're seeing through each company. And you tend to start with smaller numbers, and a lot of companies still play in that rare disease kind of unmet need space where it's smaller number of patients even when you get to commercial. So that oomph, that push for the vendors is just getting there now. As Randy and Amy said, as more companies kind of speak up and ask for the services.
Great. Well, let's take a minute here to pop up our second polling question, and I'm going to throw a little bit of gas on the fire here and say, how would you encourage, or what would you do or say to a supplier that is hesitant or is giving you kind of pushback as far as how material is supplied? And with that one, let's start with Fareed on that.
Yeah, the argument is really what keeps us pushing in the industry in general. It's the promise of this new modality. It's the promise, of a one-time, cure, and it's the potential for increased volumes as we get into as the industry in general gets into diseases that are more prevalent. Pushes in solid tumors, in oncology, and even other areas where there are products currently available. So do you want to get in on the first floor and kind of start to build that understanding and that reputation along with the industry, or do you want to wait and give that opportunity to somebody else?
Interesting. Randy, your thoughts?
It's very dependent upon the material. I mean, so you have some materials which you can find a partner to fill a set of bags. I mean, I think that's something that we have found is that there are plenty of partners out there who are willing to work with you, assuming that it's a material that they can get their hands on, that that they can fill and make appropriately, manifolds or whatever the case may be for your system. So that tends to be, I think we're in pretty good shape. The issue tends to be more where you have these unique components where you're trying to, for the first time, utilize this type of material in a manufacturing process. And there, you're working with that vendor who is not at all concerned about your formatting needs, right? They're just concerned about getting a GMP material made. And so that's when the challenge is much higher. When we find this all the time. We're working with our vendors to tell them how to become mature in a manufacturing supply space, in that environment, as opposed to the clinical supply, the early development space. And so that's where we find the biggest challenges. So I think it depends upon the type of material you're talking about.
Okay. And Amy, final thoughts on this question?
Yeah, I mean, of course, I think I agree again. I think that the biggest issues are with some of those unique components. I think there is a lot of interest in cell therapies, and vendors are definitely getting on board. But depending on the process you're working on, there's a lot of different needs. So we can think about traditional T-cell therapies, like things like activation reagents can be difficult. And then as we are now as a field looking at things like gene editing, et cetera, all those small reagents such as mRNA, those small volumes that you might be putting into the system, that's a whole nother challenge. And we're really just beginning to explore that space and what that means for supplying it into this larger core process. So a lot of challenges will be arising, I think, in those fields as well.
Wow. Great points. And it sounds like there's sort of an undercurrent of sending the message to suppliers in this space that think about not only what you're supplying, but how you're supplying it, and making sure it's compatible with the systems that you're supplying. So great. Let's take a step, I think, beyond this and think about implementation and talk a little bit about ROI. And there, in the context of there being an investment to close, integrate, automate these processes, how do you look at ROI, and how do you look at potential trade-offs when you think about closing these processes? And I'll start with Amy here.
Yeah, I think one of the immediate returns here is being able to downgrade your manufacturing space is something that we think about a lot. The more that we can take out of grade B and hoods and operators' hands and force it into grade C or lower, it really helps the manufacturing space, the space planning for your manufacturing suite, etc. So if we can get these ancillary materials in a condition where they were already developed in a very clean and robust manner, then it really helps integrate that into our larger core process that we're hoping to get into a lower grade suite. And so that's something that we think about often.
Okay. Interesting. Fareed.
Yeah, I mean, when we're talking about return on investment for automation and process closure, it again links to robustness. I think it depends on what stage you're in within that R&D life cycle. If you're still in that clinical space, and especially early phase, it's a lot more important for you to you're judging that return on investment by robustness of the process, so manufacturing failures, etc. And then that release cycle time. As you get closer to commercial, and certainly when you're in the commercial space, the financial aspect, or the cost of goods becomes, almost as important there. So those two would be the parameters that we would judge the success of an automation / process closure by.
Interesting. And as we see the poll results pop up here, about integrated system or separate sources, and think about the implications of that with some of the discussion we've had around working with vendors. It's interesting. It's almost a 50-50 split there. So thanks for everyone that responded to that poll question. Randy, I'll pass this one to you for your thoughts on this.
Yeah, I can't even tell you, the ROI starting earlier with automation is huge. I mean, if you talk to our manufacturing folks and say, what is the primary driver, if you will, in the economics of our manufacturing space, it is the number of people we have in our operations. And because we started early and have a process, our first processing with the BLA and approval, is an earlier process, very manual, lots of folks involved with producing it. I mean, our whole network strategy is how many plants can we open up across the world as fast as possible, because we need so many plants and so much space to produce these materials, right? So right. The return on investment for automation, especially if you can get it in early, is huge. The other aspect I will say is get that in before your BLA. If you're trying to do these modifications after your BLA, that is it's a huge challenge, right? I mean, that so to me, the ROI, we now have some of these cases where we can compare, and I would love to do a analysis of some of our some of the early releases of the CAR-T drugs on the market to really show the difference that can be had in terms of cost of COGM, if you will, for production, if we had been able to wait additional year or whatever the case may be to get these automated systems on board. It would have made a huge difference. So we are now incorporating them after the fact, and again, because of that, it's just a much harder job for a regulatory group to get these through the agencies.
Okay, great. So we got a question from our audience that I think is really pertinent to what we're talking about right now. Are there strategies to design clinical processes in a way to make them simpler to automate or close as the demand scales? Randy, I'm going to start with you on this one to respond to that.
Okay, I'm going to assume that we're talking autologous. I don't know from the question whether that's really true. I think that let's just start with the not. So if we're talking about allogeneic and so forth, then I think the strategies that you have are not too significantly different than you have in the protein industry, right? You have to understand the scaling of your bioreactor, the scaling of your downstream systems and so forth for larger scale processing and so forth. So I'll just leave it at that for the allogeneic space. For the autologous space, I think that what I would say is, I think I said this before, have a strategy, have an automation strategy in mind for your manufacturing space. And I know that's very difficult if you're a startup or you have a new concept or something like that. That's very difficult to do. But if there's any way that you can have a strategy for what you expect the automation to look like in the future, right? Take those concepts, utilize them in your as soon as possible in your clinical space. Example might be, you're interested in a all-in-one type production system for your process. Figure out how you can get to those systems, use them early on, understand them, because again, the advantage we have in the autologous space is you're doing a scale out, not a scale up. And so that automation that you use in that core processing anyway, and ancillary space as well as appropriate, is the same automation that would be used, right? Especially in the core process. Ancillary actually becomes more difficult because I have the ability to produce lots of material. It's not very effective in that early clinical space to be producing lots of those material, right? But still, I mean, if I have those platforms, if I can create those platforms and the understanding of what that manufacturing concept looks like, I can use those in my earlier development, and the transition into that later stage will not be as difficult.
Okay. And really quickly, I do want to encourage our audience to consider questions. We are going to spend the last probably 10 minutes of this webinar opened up for questions. So please consider what you'd like to ask the panelists and submit in the chat box there. Amy, your thoughts on this?
Yeah, I would say an important strategy, which might seem a little obvious, but maybe it's not obvious to everyone, is to invest in your development, invest in your process development team. Let them do this work, because a lot of times, especially at smaller companies, you have this great idea, you have this great therapy that you want to bring to the market, but not enough is invested in your process development. So allow them to recommend the equipment that they think that they need to automate that. Let them explore those automated equipment, and let them do the renditions of this process to really bring the most robust process into your earliest phase trial, I would say.
Okay. Fareed?
Yeah, I mean, just to continue on Amy's notes, the question was, how do you design processes, early stage processes that make it easier to transition into that larger scale later? I would flip that around and say, think about that process closure, think about that automation, think about the digitalization of the data as early as possible in your process, so that in the autologous space, it's a scale out, not a scale up, so that you're ready to roll out that additional capacity whenever it's available, and not wait until the end. The process development team, as Amy said, invest in that, invest in knowledge of what it takes to industrialize and commercialize, or even to bring to a GMP manufacturing a process that was developed in R&D, by either developing the skill sets within the PD team or embedding people that would be responsible for that GMP transition a bit later down the road. You know, it's simple things such as when you're selecting your platform, you know, equipment, think about connectivity. There are a few pieces of equipment that are available in the autologous cell and gene therapy space that you're not able to connect to a network, meaning you can't extract information from them, which makes it that much harder to digitalize, you know, later on. So think about it holistically.
Okay. And while I'm continuing to roll through the discussion here with you, I think we have one last polling question that let's go ahead and pop up. But thinking about the data handling, sampling, QC aspect of, you know, closing, integrating, automating these processes, what is the impact of that when you think about how you would execute closing and automating these processes? And here I want to start with Randy.
So if we're talking about the ancillary processes, I think that's a little bit different than the core process in terms of sampling and data and information. So if we focus on the ancillary processes, I mean, typically, you know, the kinds of things that the sampling you're doing are for sterility. You know, sometimes you have some control strategy aspects that are defined by concentrations and so forth. So there's some sampling that you're going to do, and you know, I think that is typically not at this time done. There's not a lot of PAT that's typically added to ancillary processes, put it that way. So clearly, you do need to have sampling strategies that are aligned with the single-use manifolds you're using. So if you can have manifolds that already include, in fact, sampling systems, or at least are compatible with welding on, for example, sample bags and so forth, that's something clearly to have in mind. And if that can be incorporated into the automation portion as well, so much the better. Sometimes you actually want to leave yourself some flexibility, and so you don't want to restrict too much in terms of how that is done. So again, you know, I think as Fareed had said, you know, digitizing this information is critical, whether we're talking about ancillary or core. Whatever we can do to get information, and especially if we don't have to sample, that, you know, that's that much better. But as I said before, I mean, typically you will not see the kind of PAT, you know, in an ancillary system as you will within the core process.
And interesting, really quickly, you know, looking at the poll results here, you know, the responses, they echo a lot about you know, some of the concerns that you all raised at the start of this session around closing, automating, those considerations. You know, that we see process variability, we see reduction in manufacturing failures, that stepping down to Class C, Class D clean rooms, again, you know, hugely important here as well. So very, very, very consistent messages, you know, what we're hearing. So continuing on the thread about, you know, data, QC, that type of integration, those considerations. Fareed, any additional thoughts there?
Yeah, so from a you know, a process aspect, it's extremely important to have systems, equipment, materials that are amenable to, you know, maintaining closure of that process, including sampling. Just to share an example, and I'm sure that a lot of people are struggling with this in the autologous cell and gene therapy space, where again, that turnover, the turnaround time is extremely important. A lot of people are moving to BAC-T testing for sterility. BAC-T, the actual, you know, capsule needs to be you know, injected or seeded via a syringe right now. So it forces you to open that process at some point. If there is a way to actually transfer that material into the capsule in a closed manner, there's obviously a lot of benefit there, including reduction of errors that occur during that transfer. So, you know, it's extremely important to maintain that closed as well, all the way until you get to the testing lab. And that's especially important for safety tests, sterility, endotoxin, myco, etc. Automating the data and digitalizing it is extremely important to that, you know, turnaround time as well. The more that data is is digitalized, the easier it is for quality assurance to review, to release, for exceptions to be noted, but also as you get closer to, in the R&D space, but even more importantly, as you get closer to commercialization, the trending, monitoring, and early detection of changes in your process. The easier it is to go into a database that's looking for trends, that's looking for outliers, etc., the faster you react to some of these changes, and the better control you have of your process.
Excellent. Amy?
Yeah, I'm not as familiar with, you know, as Randy said, PATs, anything like that for ancillary processes. But when we think about processes, as Fareed mentioned, you know, any sort of process monitoring that you can have, and you know, that you can build a data set over time early on and kind of understand what those process changes might indicate about that process, whether it's changes in pH or DO or anything like that, would give you an early indication as to how that process is going, if there is a risk of contamination or anything like that. All of those things will help play a role in understanding, you know, what's at risk in your process, how your process is going, etc.
Great. We do have a question that came in from the audience. So looking towards or looking forward 2 to 3 years, what hurdles do you expect to overcome, and what challenges do you expect will still persist? And let's start with Amy on this.
Yeah, I think that that's an interesting question. So when I look back to the last, you know, 5 to 8 years in cell therapies, we've come a long way. You know, back then we were really leveraging devices that were coming out of clinical settings. So things like the Cell Saver 5, which is used to recycle blood in surgery. We were using that as a cell washing tool. Now, when we look across the market, there's a lot more automated devices for that process unit. Granted, there's still a lot of ranges in terms of volumes and other things that we can improve upon. We've still made a lot of advances in this short amount of time. So I think in the next 2 to 3 years, the amount that companies will be investing into automated options will continue to increase. We'll see more off-the-shelf options for bridging some of those gaps that we're currently experiencing in manufacturing and automating processes. However, the field is always changing as well. There's going to be new therapies on the market. You know, as mentioned earlier, there's more and more things like gene therapy and stuff like that, which are bringing different challenges into the space in terms of automation and supply of those ancillary materials. So I expect that over the next few years, those sort of processes will also be driving what sort of things are coming on the market. And so I'm very optimistic in terms of what's going to be coming out of this next rendition of cell therapies.
Great, great. Fareed, your thoughts?
Yeah, I'm along the same lines as Amy. I think 2 to 3 years from now we'll be in a much better place. I don't think that we would have addressed all of these issues. I think a lot of them are moving in parallel with each other. So it's not like we're going to get one done and you know, a few of them remain. Process closure, smaller scale systems, components that work for the cell and gene therapy space, automation, integration, etc. All of this is going to work, is going to start to move in parallel. And you know, my time frame, or the time frame that I foresee, you know, addressing these is probably in the 5 to 7 year period, as opposed to 2 to 3 years. Again, I foresee some significant developments by then, but probably not closing out everything. I do agree with the point that Amy made here, and that's the fact that the field of cell and gene therapy, again, focusing on autologous, will also change and will most likely go towards smaller volumes, smaller apheresis, leukapheresis material from the patient themselves, so that they don't have to go through long, arduous apheresis processes, smaller vector volumes for that transduction, etc. So it'll needs to be adaptive as well.
Okay, great. Randy.
Yeah, I think to just continue the theme, I believe that the immediate challenges with the first modality we have in CAR therapy and cell therapy modalities we have currently are what we're going to resolve in the manufacturing space over the next 2 to 3 years to make it more efficient, more affordable, more accessible. I think that's something that we are now taking on as a challenge and understand it and are getting the vendors on board. The challenges that we have is that the modalities are going to change so fast, our heads are going to spin, right? Because there's going to be the next version and the next version, the next version, and you're always dealing with that next vendor who has that unique material or that unique methodology that you've got to figure out, how do I turn that into a GMP manufacturing process, right? That to me is the ongoing challenge that we will have as this field continues to develop and have introduced so many more new and effective modalities.
Yeah, and that's an interesting point, because I think and everybody, I think you guys are spot on, and I think that's the industry working with organizations like us, like Invitech. They're going to continue to push us and others like us to continue development to really push the edge of manufacturing and be able to support where these companies are going, and really have the opportunity to kind of lead the way there. So I want to, and that was a fantastic wrap up, and thank you to the audience for that question. It was perfectly timed. We are at the end of our time period, so what I'd like to do is thank the panelists and thank Facilitate as well.
Thank you, David. And I'd like to extend a huge thank you to all the panelists. We've covered some really critical points today when it comes to automating ancillary materials. So just a quick note that this webinar will also be available on demand. So if there's anything that you need to refer back to, look out for the link for the recording in your inbox later today. And to keep up to date with more things like this and resources and webinars, visit Facilitate or the Facilitate Exchange. Thanks everyone again, and see you all soon. Have a good afternoon.
Thank you, everyone.
Thanks.
Thanks all.